Hello friends, welcome to the Plant Science Forum. In another video, we had discussed about the photophosphorylation, C3 Kelvin cycle, C2 photorespiration and C4 cycle. Now, I will be talking about the another consequence and adaptation occurs in the dark reaction. Among them, now we are going to discuss CAM pathway, crassulacean acid metabolism pathway. Now, while discussing this topic, we will try to answer these questions. What happens in normal conditions? Carbon dioxide concentrating mechanism. Why called as CAM pathway, crassulacean acid metabolism pathway? Why CAM pathway has developed? Where does it happens? How does this pathway lead to the carbon dioxide fixation? Now, what are the consequences and significance of CAM pathway? This is a very general representation for what happens in photosynthesis in normal condition. Photosynthesis consists of light dependent reaction and light independent reaction. Light dependent reaction involves light mediated excitation of electrons from chlorophyll, where electron vacancy which is being created is fulfilled by electrons liberated by oxidation of water molecules, and oxygen is byproduct of the reaction. The ATP and NADPH produced by the light reaction is utilized in the next dark reaction or light independent reaction, that is Kelvin cycle. In the Kelvin cycle, atmospheric carbon dioxide is absorbed and incorporated as the carbohydrates. Here ADP, inorganic phosphate, and NADP is byproduct, which is now enters in light reaction to get reduced. As we had discussed and understood that for the synthesis of carbohydrates by fixing atmospheric carbon dioxide, RUBISCO enzyme plays a very important role. In nature, plants are present in very diverse habitat where principal pathway for carbon dioxide fixation, that is Kelvin cycle, could not operate effectively. So, plant adapted to their present environment by variety of processes that delivers atmospheric carbon dioxide to the Kelvin cycle. In such adverse condition, Kelvin cycle operates in a protected environment deep inside the cellular arrangements. So, these adaptations are C4 cycle or CAM pathways. Now, plant has developed this mechanism for concentrating carbon dioxide around RUBISCO for operating Kelvin cycle for fixing carbon dioxide and subsequently synthesizing carbohydrates. So now in this talk we are going to discuss one of the adaptation, that is CAM pathway, crassulacean acid metabolism pathway. Now why this called as a CAM pathway? Because after the name of plant family Crassulaceae, in which scientists first discovered this pathway. Despite its name, crassulacean acid metabolism is not restricted to this family only, that is Crassulaceae family. Along with this Crassulaceae family, it is also found in cacti and many euphorbia species, as well as pineapple, vanilla and agave also. So this is overall where CAM pathway actually reported. So here a pineapple, vanilla, agave, these three are exceptional plants which we have discussed here, and rest other, these are a Crassulaceae family members. So this pathway is not only restricted to the Crassulacean plant family. Now why CAM pathway was developed by nature? Because CAM pathway allows plants to survive in inhospitable climate where water loss is a major limiting factor for plant growth. Now this CAM metabolism eliminates evapotranspiration through the plant stomata, which usually opens in the daytime. Evapotranspiration means water loss through a gaseous exchange. Just like C4 plants, CAM plants also in CAM plants also RUBISCO is protected from high temperature and does ultimately from oxygen. So this means that CAM pathway in CAM plants the RUBISCO enzyme doesn't undergo a photorespiratory process, so it is protected by oxygen. Now this CAM pathway, it happens in mesophyll cells. The CAM mechanism is similar in many respects to a C4 cycle. In C4 plants, formation of C4 acid in mesophyll is specially separated from the decarboxylation of C4 acid and from refixation of resulting carbon dioxide by Kelvin cycle in the bundle sheath cells. Whereas in CAM plants, the pathway operates in mesophyll cell as we have said earlier, and formation of C4 acid is both temporarily and spatially separated. Temporarily means by time, spatially means by space. This is we are going to discuss in next slide. As you can see here, this is a general C3 pathway in which carbon dioxide enters into the plant body in leaf via stomata, and this carbon dioxide gets fixed in the Kelvin cycle in chloroplast of mesophyll cells. Now in C4 plants what happens? Carbon dioxide is received by mesophyll cell and converted into C4 acid. Now this C4 acid get decarboxylated in the bundle sheath cell, where actually carbon dioxide is fixed into a Kelvin cycle and ultimately forms a carbohydrate. Here you can see a mesophyll cell is a different entity and bundle sheath cell is a different entity. So carboxylation, formation of C4 acid, and decarboxylation, fixation of carbon dioxide, this is separated by a space. One happen in mesophyll cell and another event happens in bundle sheath cell. So this is a spatial separation. Now let's see a CAM plants. Now just look at the CAM plant. Here carbon dioxide enters into a mesophyll cell and it will form a C4 molecule or C4 acid in the night time, whereas this C4 acid get decarboxylated and form carbon dioxide, and this carbon dioxide get fixed by a Kelvin cycle in the daytime. So here there are two factors, one is time, that is night and day, another is the space. So one thing, that is carboxylation and storage, it happens in vacuole, whereas the carbon dioxide fixation it happens in chloroplast. So there is a special separation also. That's why it is said that in CAM plants the complete process is spatially and temporally separated. Now let's have overall idea about CAM pathway. At night, carbon dioxide is captured by a PEP carboxylase. Just look at here, phosphoenol pyruvate carboxylase, and this is carbon dioxide which is captured by enzyme PEP carboxylase in the cytosol, and the malate that forms from the oxaloacetate product is stored in the vacuole. It is stored in the vacuole. During the day, the stored malate is transported to the chloroplast and decarboxylated by NADP malic enzymes. The released carbon dioxide is fixed by a Kelvin cycle, and the NADPH is used for converting the decarboxylated triose phosphate into the starch. So this is an overall representation for what happens in CAM pathway. Now let's see in details. CAM plants such as cacti achieve their high water use efficiency by opening their stomata during the cool desert nights and closing them during the dry days. during the hot dry days. So closing the stomata during the day minimizes water loss, but because water and carbon dioxide shares the same diffusion pathway, that is through the stomata, carbon dioxide must be taken up at night. Carbon dioxide is incorporated via carboxylation of phosphoenol pyruvate to oxaloacetate, which is then reduced to malate. The malate accumulates and is stored in the large vacuoles. The accumulation of substantial amount of the malic acid is equivalent to the amount of carbon dioxide assimilated in night time. With the onset of day, the stomata get closed, thus preventing loss of water and further uptake of carbon dioxide. The leaf cell deacidifies as the reserves of vacuolar malic acid are consumed. Decarboxylation is usually achieved by the action of NADP malic enzymes or on malate. Because the stomata are closed, the internally released carbon dioxide cannot escape from the leaf, and instead it is fixed and converted to carbohydrates by Kelvin cycle. The elevated internal concentration of carbon dioxide effectively suppresses the photorespiratory oxygenation of RUBISCO and favors carboxylation, and by this it means the C3 Kelvin cycle. Now again let's revise what I said right now. Here carbon dioxide enters into the mesophyll cell in the night time. Now this carbon dioxide mixes with the water molecule and forms bicarbonate ion. This bicarbonate get attached to phosphoenol pyruvate under the enzyme phosphoenol pyruvate carboxylase, and it will form a C4 acid oxaloacetate. Now this oxaloacetate get converted into malate, and malate is converted into malic acid and stored in the vacuole in the night time. Now this phosphoenol pyruvate it forms by the starch starch converted into triose phosphate, and this will form a phosphoenol pyruvate. Now what happens in daytime? In the daytime stomata is closed, so the malic acid which is stored in the night time in the vacuole, it get converted into malate and it enters into the cytoplasm. After the cytoplasm it enters into the chloroplast. In the chloroplast malate is decarboxylated and converted into pyruvate, and this pyruvate again converted into a starch from it has came. And after the decarboxylation the carbon dioxide which is released, this carbon dioxide get fixed by the Kelvin cycle, because as the growing concentration of carbon dioxide because of decarboxylation of malate, this carbon dioxide cannot escape the plant because the stomata is closed. So ultimately the concentration of carbon dioxide get increased around the RUBISCO enzyme in the Kelvin cycle, and because of which it favors a carboxylation process, and ultimately it forms a carbohydrate molecules. Now what are the actual significance of CAM pathway? Now this CAM mechanism enables plant to improve water use efficiency. Typically CAM plants loses 50 to 100 gram of water for every gram of carbon dioxide gain. Now these values, if we compare it with the C3 and C4 plants, then it is much less, and the values of water lost by the C3 and C4 plant is very much higher as compared to CAM plants. Thus CAM plants have a competitive advantage in dry environments. The disadvantage of CAM plants is that they often have low photosynthetic capacity, low or slow growth, and low competitive abilities because of their photosynthetic rates that are limited by the vacuolar storage capacity and by greater ATP cost, similar to what happens in C4 species. I hope you might have understood the topic. If you like the video, please subscribe to the channel. Thank you.